I am pleased to introduce Mike Lowe for today's webinar. Mike, you want to pull up your slide deck? Mike got his bachelor's in marine fisheries from Texas A&M University, Galveston. He got his master's in fisheries from Auburn University, and then his PhD at University of Southern Mississippi. He's did two postdocs, one at Woods Hole, where he analyzed long-term data sets, looking at trophic interactions and sort of some fish habitat. Then he came back to the South, LSU Agricultural Center, where he did some habitat and stock assessment work in the Gulf of Mexico. And he is currently stationed at the Hammond Bay Biological Station, working for USGS. And we will hear, I think we're seeing your notes. Maybe swap those, swap displays there, Mike. He's going to be sharing some work that he's been doing with us on Buffalo Reef within Keweenaw Bay, Lake Superior, looking at the impacts of the stamp sands on the habitat for native fishes, including Lake Whitefish, which is, of course, the hook to get him into this webinar. So we're excited to hear what Mike's going to share with us. Talks about 45 minutes or so. And again, we really hope that people stick around for questions. We're really happy to see so many of you back after our summer hiatus. And thanks again for volunteering, Mike. The stage is yours.
All right. Thanks, Bo, and thanks for inviting me to do this. It's I've always enjoyed these webinars, so it's nice to be a part of it.
Fantastic. Thanks.
So before I jump into the real sciencey part of this webinar, I wanted to give a little background on Buffalo Reef because it really is it's the key driver of why we are doing this work, but it's also a really interesting part of the story. So in this map that I'm showing here, this is the Keweenaw Peninsula along the southern shore of Lake Superior. It's commonly referred to as copper country because between 1850 and 1920, it was the second largest producer of copper in the world. Now, in fact, the amount of copper that was extracted during that time frame, you could make about $16 trillion in modern pennies. So while it was lucrative, it came at a cost to the environment because they were breaking down these ores that really only contained about 2 to 3% copper. So 90+% of whatever it was they broke down was waste material that had to go somewhere. And they made multiple deposits all over the Keweenaw Peninsula. One in particular is just north of this embayment that you can see here at the star. That star is Grand Traverse Bay. And just north of that is Gay, Michigan. There was a big stamp mill there. It's estimated that by 1932, they had deposited about 23 million metric tons of this waste mining material that's commonly known up there as stamp sand. Now, this is that same Grand Traverse Bay. I've rotated it now so that north is to the right, and that's north, and to that direction along the shoreline is the original stamp sand deposit. As I said, by 1938, there was about 23 million metric tons of sand deposited right there outside of Gay. That over time has eroded and migrated, drifted down the shoreline. And that's kind of what you can see here in this picture. So if you look at the dominant shoreline from right to left, you can see this large, wide swath of grayish black sand until you get down to the harbor jetty that was built in 1958 as part of the Safe Harbors Act. And then on the other side of that, you can see the natural beach. The jetty was just built to protect the harbor. They didn't anticipate or they didn't know that one day it would actually be protecting this natural beach habitat along the southern shoreline. So this is why we are here today. This is Buffalo Reef. It's a 22 hectare cobble and limestone reef that extends from the shallow near shore zone at about less than a meter of water out to about 30-35 meters. Um, there's a few key features that you can see in this high resolution bathymetry data that we've used to build this 3D model. Um, so the first thing are these large sand dunes, underwater sand dunes up here on the north side of the reef. Um, these are mostly stamp sands, and you're talking about underwater sand dunes that can be 5 or 6 meters tall in some areas. The next feature of the reef that's important to note is the trough. This was a dugout channel that for decades was capturing the stamp sand as it flowed out into the bay, and probably protected the reef for a long time. Because we'll talk about in a few slides, it's not until the late 80s and early 1990s that tribal fishermen started noting that there was stamp sand on the reef. And then the two main features of the reef are these bathymetric humps that come up near the middle of the reef. Um, I don't have any depth for scale on here, but the apex of both of these humps is about 7 meters deep. So for a visual of how much that beach has changed, how much that shoreline has changed over time, on the left we have a picture from 1952 that was taken by Bob Regis, who's a professor up in the Upper Peninsula. His family has owned a cabin up there for since the 30s. Um, and this is approximately on the right, this is approximately that same swath of beach as it looks today. Not only is it covered by these massive amounts of stamp sand, the beach profile is radically different, and that extends out into the water too. Because if you were to step off of this water line here, you would probably fall in above your head. It's that steep. We've done zooplankton tows out there, and you can be doing a zooplankton tow and stick your hand out off the side of the boat and grab a handful of sand while you're doing it. It's deep enough for that. So not all of the stamp sand has been confined to the shoreline. In fact, most of it, about 12 million metric tons of that original 23, is estimated to have washed out into the bay, and now is covering about 60 to 70% of the reef area. So what we're looking at here on the left is an interpolation that Peter Esselman's group did using side scan sonar and ponar grabs to give us a more current understanding of the spatial distribution of stamp sand on the reef. So it grades from white, or the cooler colors, are the low concentrations, to the dark reds being the highest concentrations. And you can see that there's a clear pattern that the near shore areas around Buffalo Reef have been heavily impacted. What's difficult to see too, though, is that we are seeing some pretty considerable buildup in the center of the reef around those humps. So to give you an idea of what this looks like, I have this image on the lower right, and this is just a microscope image when we try to assess how much stamp sand's in a subsample. This is kind of what it looks like. So you have the quartz sands, which are these kind of rounded, clear, kind of translucent grains. And then you have these dark, opaque, kind of jagged grains, and those are your stamp sand. Now, what we know is that these grains of stamp sand still contain appreciable quantities of copper in them. Okay, and so this is a paper that this figures from a paper that I just got word today has been accepted with minor revisions. It it shows the relationship between percent stamp sand in the sediment or in the substrate versus the amount of copper that's in the substrate. So here, the take home is that as you get above 30% stamp sand, you start encountering copper concentrations in the substrate that are potentially harmful to aquatic life. So it's at this point that I have to stress that I am not a toxicologist. I'm simply a fish ecologist that's trying to help understand what the long-term effects of stamp sands are, and how that's affecting fish production at Buffalo Reef. You know, so it's safe to say that fish have evolved a sophisticated mechanisms for maintaining copper homeostasis. Um, and then just acknowledge that repeated exposure to high levels of copper can overwhelm these mechanisms and have strong negative effects on fish health and survival. So this is just kind of an adaptation from a figure in another paper I've brought into this talk. It's an overly simplified conceptual model. The arrow captures the decrease in survival as the increase in waterborne concentrations of copper increases. So now we're talking about aqueous copper concentrations and not necessarily the copper in the substrates. Um, okay. And then this arrow will serve as kind of a separator between what's going on inside the fish as copper concentrations increase, and that's going to be the part below the arrow, and then above the arrow will be more of the behavioral and population level responses that we might expect to see. And then down here, I've got two arrows that show kind of the naturally occurring range of copper in both seawater and freshwater. So at low levels that we see in the natural occurring range, we see levels that are essential for biological function. Um, that doesn't mean that we can't reach concentrations that are somewhat of an impediment to these animals, but they can typically acclimate. But as in some cases, these higher levels of naturally occurring copper can block a species' ability to smell certain habitats. A good example is salmon during their spawning migration. you know, from there, things kind of escalate quickly, and these increases in copper can cause various forms of internal physiological stress. With the most immediate response is for the fish to avoid those areas with high copper. But in species that have a limited home range or as a habitat specialist, complete avoidance may not be possible. And that's where we start to see these demographic changes during the early life stages, development rates, growth rates, changes in reproductive output. And then as you increase copper in the water, the fish are unable to regulate their internal copper concentration, and their systems start to break down. There's an ionic imbalance in the fish, and then ultimately there's cardiovascular collapse. So, and this is where we start to see kind of direct mortalities related to copper toxicity. Any further increases beyond that, and you start to see, local extirpation of the population. Um, Sorry, my screen blanked out on me for a second. I forgot. I couldn't see where I was. There we go. Um, so lastly, it's important also to understand just how potentially toxic the waters are around Buffalo Reef. Um, so this last arrow that I've added down here on the bottom just kind of shows the ranges of copper concentrations that we found in interstitial waters on Buffalo Reef, where both lake trout and lake whitefish are spawning. Okay. Um, Up at the top on the right, I've got an image. So to do this, we had to use specially made autonomous water samplers that were diver deployed They could insert these sampling tubes down into the interstitial spaces, and then we could take a water sample at a predetermined time interval to give us an idea of what are the short and long-term changes in copper concentrations in these interstitial spaces. And the take home here is that not all of it is incredibly toxic to lake trout and lake whitefish, but there are some areas of the reef that they're most likely, if they deposit embryos there, they're not going to survive. So the other reason we're here is because historically Buffalo Reef has been recognized as one of the primary producers of lake trout in lake or sorry lake whitefish in Lake Superior. Um, This figure down on the bottom right shows the commercial landings of both lake trout, which are the black bars, and lake whitefish, which are the gray bars, the gray vertical bars. For the years of 1985 through 2018, these data were supplied by Bill Mattis from Glyphwick. Um, and then there's a solid line that's above the bar plot that shows the proportion of the catch for 1842 for the 1842 waters of Lake Superior that were captured within 50 miles of Buffalo Reef. And that 50 miles is shown in that map to the upper left with that dark gray area. It's a pretty big area. And as you'll see in a couple of slides, there's a lot of reefs in there that could be contributing, but previous studies by Bill and others kind of indicated that Buffalo Reef was a primary contributor to fish or to the fisheries in that area. But the other take home from the bottom right hand figure that I also want to point out is you could see that around 1992, 93, there's a big drop in terms of the landings of Lake Whitefish in that 50-mile area, the 50-mile buffer. And that timing really overlaps with when tribal fishermen started noting buffalo or stamp sands kind of inundating the shallow areas of Buffalo Reef. So I don't think we need to get into the life cycle of Lake Whitefish for this audience. I'm fairly certain most folks are familiar with it. But what I do want to point out and highlight is that the reef, and in this case, Buffalo Reef, is the template on which all of a lot of these critical population dynamics are occurring. So the spawning, the egg overwintering, development, and then the hatching to the sac fry and the larval stages. And even then, it still remained the stamp sand pile is still connected to these nursery habitats where the sac fry and the larvae are going because it's drifting down into the lower part of the bay, which is widely recognized as the primary nursery habitat for Buffalo Reef or for whitefish produced at Buffalo Reef. All right, as I promised, we're going to get into a little bit of science now. So the first my first introduction to Buffalo Reef was working with Bill Mattis and Ben Michaels and Andrew Muir and Tom Binder to develop an acoustic telemetry project to identify where on Buffalo Reef lake trout and lake whitefish are spawning currently. This project started in 2019. We tagged 41 lake trout or 41 lake whitefish uh, as they came onto the reef to spawn. And then we have had an array out there every year since 2019 to look at, where these fish are spawning relative to the stamp sand distribution. Then in 2022, we started up a new project, and I'll describe the projects a little bit in more detail in the next slide, where we added between 2022 and 2023, we added another 250 um, uh, lake whitefish that were tagged with electronic transmitters. Um, in here in this picture, you can see us. We're actually working from one of the tribal gillnet tugs up there. They would bring in the fish. We would monitor their health in this round circular tank and tag the fish, and then monitor them some more, and then we would release them. It really did take a team effort, and it's a lot of fun working with the tribal fishermen on this. So as I said, the first project, the first telemetry project was a fine scale VPS study. The goal of this was to determine where on Buffalo Reef these fish were spawning and how that relates to the stamp sand on that's inundated the nearshore areas at least. This is just a depiction of the VPS array on the bottom right. So each one of those triangles is a receiver, and we have approximately about 500 meters of spacing between them. The only place where that's not true is in the center of the reef, where those two humps are. Because of the differences in bottom height, we put in more receivers in that area just so that we didn't have any acoustic dead zones where the receivers couldn't communicate with each other. And then that yellow circle that you see in the middle, that's kind of a relative location for where the fish were when we caught them during the tagging for the tagging. The other project, the other acoustic telemetry project, is more focused on kind of broad scale patterns of reef usage. So the state of Michigan funded a project to look at uh, what reefs were contributing to the fishery in Keweenaw Bay. And the idea was to go out with the commercial fishermen again and tag fish in the areas where they would typically fish them in the springtime. Um, Obviously, the emphasis is to understand how important is Buffalo Reef today in terms of fisheries contribution. But we use the Goodyear 82 document to kind of get an idea of where the reefs might be located, and then we conferred with tribal biologists and state biologists to determine what reefs were worth putting receivers on. So this is a project that started in 2022 and will end next year. But we've got some pretty interesting data from that, and I'm going to talk about that first. So, like I said, we tagged 80 fish in 2022. Um, and we were shocked to see. So this is just to give you an idea of what the layout is here. So on the left, on the y-axis, we have the various stations that corresponds to the figure on the upper right-hand side. So if you're moving from the tip of the Keweenaw Peninsula down towards the end of the bay, you get to um, you get down here to what is it, Sandstone Point, right here in the middle by the inn on the station. And then as you continue down this plot, you start moving over to these reefs along the southern shoreline of Keweenaw Bay. So that's how this is oriented. Along the x-axis, we've got weekly increments to give an idea of when there were fish present. So this is what you're seeing in the dot plots are the total number of unique fish that are visiting these sites on a daily basis. Okay. And then the gray bar in the middle is the suspected spawning window for fish for Lake Whitefish in Keweenaw Bay. And then I've highlighted Buffalo Reef with the yellow strip. And the thing that really leaped out to us is that we tagged 80 fish. We had our other 41 fish that we had tagged in 2019. Those tags have been active through this fall. They should just be now coming offline. Um, but the thing that leapt out to us in this was that, well, we're not getting any whitefish on Buffalo Reef. Um, and we know that that's not an accurate reflection of reality based on the VPS study. And I'll show you those slides here next. So what we determined was we were being a bit too conservative in where we placed our receivers. We were a bit worried about ice and receiver loss. So we think that this year that we were just a bit too deep. We moved it, and the following year, And we do start to see, so these are the fish. This includes the 200 or the sorry, the 170 170 lake whitefish that we tagged throughout Keweenaw Bay in the summer of 2023. Now we do see fish showing up at Buffalo Reef, but it doesn't appear that important. I think that our maximum number of unique fish that were on the reef at any one given time during that window was six individuals. So six out of 250 at this point, which again, or no, sorry, 291. Um, so it does suggest that the fish are not using Buffalo Reef like they used to. And in fact, that if you look at this plot, particularly with the year two data and the year one data, it's these reefs that are north of Buffalo Reef that appear to be visited the most by lake whitefish during the spawning period. And then the other interesting pattern that kind of jumps out too is that these reefs on the southern side on the southern edge of Keweenaw Bay don't appear to play much role in terms of whitefish reproduction for Keweenaw Bay. So in terms of the fine scale positioning, um, as, as recently as 1998, Gliffwick and tribal fishermen were sampling gravid, ripe running lake whitefish females in the shallow areas of Buffalo Reef that's highlighted by this box. Okay. Um, this area of the reef tends to be shallower than 5 meters. There's a lot of cobble habitat, naturally occurring cobble habitat. But as you can see from Esselman's stamp sand interpolations, it's pretty inundated with stamp sands. And you can see that if you're out there in a boat, you can look down and you might occasionally see the top of a rock or a little cobble field, but it's mostly just covered with sand at this point. So when we got the VPS data back in 2020, we were expecting them to still be going up into that shallow area to spawn, and that just wasn't the case. In fact, it looks like they're completely avoiding that historical spawning area at this point. And the hot spot for their activity appears to be out in about 13 meters of water, which, if that's where they're spawning, and we think that it's pretty consistent with spawning behaviors, what we're seeing, if that's where they're spawning, then it's likely the deepest recorded spawning that we've seen yet in Lake Whitefish. It's worth noting too that this pattern has been consistent in 2020, 2021, 2022. I don't show it yet because I just got the data back and haven't had a chance to really dig into it. But so far, it looks like the exact same pattern is present again in 2023. So, the take home is that we've got a few lake whitefish that do appear to be continuing to spawn on deeper parts of the reef, but they are not venturing anywhere near these highly impacted habitats. So at the same time, as starting in 1996, Gliffwick started monitoring the juvenile abundance or the age zero abundance of Lake Whitefish in that nice, pristine, white, sandy area that's south of the harbor entrance that I showed you on a previous slide. And you can see this. So this figure here on the right is kind of a summary of those catches, showing the young of the year density as a function of number per square kilometer. The solid line gives an indication of the amount of effort that was put in each year, um, which captures the water volume sampled. And you could see that up until about 2008, it was kind of a typical response where we have these boom and bust years, but there were a lot of booms. There were a lot of strong cohorts showing up until about 2008. And then after 2008, they stopped seeing those strong cohorts. And occasionally they would get a few fish in their seines. Now we dug into this data and talked with Ben and Bill. And one of the things that came out of this is that they had a set location at which they would do their seine hauls. And we noted that the stamp sand maps were starting to show that there was stamp sand moving around the harbor entrance and was actually starting to fill in in this area where they were doing their sampling. So we were concerned about what that meant for the quality of the nursery habitats as well. So we started a comparative study with Glyphwick and KBIC in 2021, where we replicated their early survey work, but we compared it with biweekly seine samples at four other uh, nursery areas that are known to produce a lot of or thought to produce a lot of whitefish. Um, so the so on the upper right hand, I'm showing you the map of where those five nursery areas are. Starting from the top, we have Great Sand Bay on the western side of the peninsula. Then we move over to Bay de Gris, which is up at the northern part of Keweenaw Peninsula. Grand Traverse Bay, which is essentially the nursery area for Buffalo Reef. Little Traverse Bay, which is another really nice white sandy embayment that's around the point just south of Grand Traverse Bay. And we do see small quantities of stamp sand coming around that point and into that bay currently. And then Big Bay, which is all the way over here on the southeastern side of Keweenaw Bay. Um, this figure here on the bottom left kind of gives an idea of what the catch per unit effort is. So this is just the number of fish, the average number of fish per seine haul in each of the sampling years so far. So 20, 21, 22, and 23. We do have 24 data, but haven't had a chance to work it up as we're still waiting on samples from Glyphwick and KBIC. Um, but the main point here is that even though we did see lake whitefish, age 0 whitefish present at the Grand Traverse Bay Nursery, it was almost universally the lowest CPUE of the five sites. And I think in 2021 and 2023, we had fewer than 10 fish caught in 27 seine samples in each year. So not a lot of production based on this. Um, if you look at that odd looking figure in the over Little Traverse Bay, that's the LTB just to the right of the yellow bar. We had that year, which was 2022, we had just a massive cohort of lake whitefish in that bay. We had seines we had two seine hauls. One had 2,300 and the other had probably, I think, 800 or 900 individuals. And we decided not to do a third seine haul after that. Um, so for some reason, Little Traverse Bay was really productive that year, but not very productive in the other years. So a big component of the work that we've been doing is focusing on otoliths to quantify potential differences in habitat quality at Buffalo Reef when you compare Grand Traverse Bay, particularly hatch date distributions and growth rates to these other nursery areas. Um, as most of you know, aging lake whitefish can be difficult. And we spent a lot of time conferring with Andrew Muir and looking at Aaron Dunlop's work, following it pretty closely. And we've used that to kind of revise our process over time. Katie Dawson, who's a technician in my lab, she's taking care of. She's the one that's primarily doing the aging, all of the mounting, the polishing, the aging, the different measurements, the fish identifications, and she's taking care to note, where these different checks are occurring. So you have, in conferring with Andrew Mira, we started looking specifically for two checks. One is that hatch mark that where they start laying down where they start laying down daily rings following emergence from the chorion. Um, that was really difficult to consistently identify in our wild caught fish, until we had the bright idea to look at our laboratory reared fish, and get a better feel for what that check should look like and where it should be. And then once we had that, so that's what I'm showing you here on the upper left is an otolith from a 9 day old post hatch lake whitefish that we reared from from fertilization all the way up to 9 days old. And looking at that data, when we estimate the number of rings that are potentially laid down prior to that check mark, it comes out to about 5.4 rings on average. The range so far has been about 4 to 8 rings. And that gives us a good starting point for when we're trying to identify the ring, the hatch mark location in our wild caught fish. It also gave us a good, strong indication of what our size at hatch was. And we know that it doesn't necessarily correlate to what's going on in the wild, but 12.6 millimeters at hatch was our average size for our laboratory reared fish. And we use that for or for our estimates of growth rates. And then these are, um, and I should add too that, one of the things that Katie has been working on, and it's proved to be really useful, is she's building these tiled mosaics of the full otolith, for our wild caught fish. So she's able to take these and line them up based on landmarks, and she can focus in and out to get the best view of the daily increments. And then she can stitch these images together and build this full tiled mosaic that allows us a better view of the rings in some areas where we can't quite discern, subdaily and daily increments. Um, and so these two figures just kind of show the the relationship. The top one shows the relationship between otolith diameter and standard length. We have a pretty strong relationship, with R squared of about 98, 0.98. So it's a pretty good indication that we are seeing daily increment formation, and that this is corresponding to, or it does kind of drive home the point that we can accurately age these fish. Um, and then the figure on the bottom right just shows the estimated age in days post hatch versus the standard length of the fish. And again, this is the relationship that we would expect to see, that there's linear growth early on, and then growth rates start to increase pretty dramatically, once you get above 30 millimeters or 30 days old. So comparing growth rates across the 5 nursery sites. So we've got data here from 2021 and 2022. So 21 is the dark bars, 22 is the light gray bars. And then each panel represents one of those nurseries. So moving from left to right, you have Great Sand Bay all the way over to Big Bay, with Grand Traverse Bay here in the middle, which is highlighted in yellow. The take home is that our growth rates were pretty consistent among individuals within a site, and Grand Traverse Bay fish were almost always had the slowest growth of the 5 sites. So the other interesting part of this is we wanted to look at the growth rates as it related to capture date, which is now along the x-axis. You have the same kind of spatial layout, and this is just for data collected in 2022. And the interesting thing is that while we see kind of a general increase in rapid growth as you move into mid-July, when water temperatures are warm, there's lots of food. You see that you kind of see that at all 4 sites, but particularly at Little Traverse and Big Bay. It's really obvious. But when you look at Grand Traverse Bay fish, their growth rates actually slow down as you move into July. And we think that this is a prey availability issue. And just, I said earlier that we just had a paper accepted where we were looking at these at prey availability in three of the those five nursery areas. So we've got Grand Traverse Bay, Little Traverse Bay, and Big Bay. The take home is zooplankton density was incredibly low at Buffalo Reef, and we don't know why. We had no reason to suspect that the stamp sands would affect the zooplankton up in the water column, but they did. Abundances were nearly an order of magnitude lower. Taxodiversity was lower. And then that kind of translates also into the benthic prey community. So I showed this first figure here on the left earlier, showing the relationship between percent stamp sand and copper in the sediment. And when we take that copper in the sediment and we plot benthic density as a function of that, we see that there's a rapid decrease to near zero as these concentrations of copper increase. So basically what we're seeing is that in these habitats that are heavily impacted by stamp sands, copepods, amphipods, nematodes, chironomids, they're either noticeably absent or they are rare in those samples. And then this summer, we decided to shift our focus more towards larval lake whitefish for our studies. One of the things that we've been trying to do is use otolith microchemistry to see if we can determine natal origins um, among these different spawning reefs in Keweenaw Bay. We tried to do that originally with the age zero young of the year fish. We think we had still too much otolith material, so the results weren't really as clear as we had hoped. So we tried it again with larval fish, and as I'll show you in a few slides, we were pretty successful. So rather than using traditional zooplankton larval fish nets to get these guys, we just wanted to get sheer numbers. So we started using light traps and what we're calling active light trapping, where we would anchor up the boat in the middle of the night, put out these high lumen lights on each side of the boat, and then we would just dip net the larvae as they came in. We were pretty successful at this. So, and again, just acknowledging too that we understand that it's not easy to identify lake whitefish or discern lake whitefish from other chorgonids at this life stage. We're fairly comfortable with our identifications, but we're still going to submit them to Amanda to get some genetics done and confirm that they are whitefish. But anything that we're analyzing in this is whitefish. We confirm that there's a paired double row of stellate melanophores along the back. They fit within the size range, and we did see a bunch of lake white or a bunch of chorogonid larvae that were too small to be lake whitefish during this time. And so we've excluded those from this analysis. So this shows the catch per unit effort among our 7 sites that we did the active light trapping at. So again, these are color coded. So orange, the 2 orange sites are going to be north of Buffalo Reef, and then the 3 kind of greenish sites are going to be south of Buffalo Reef, with the 2 blue sites being either Buffalo Reef or Grand Traverse Bay. Surprisingly, this does not line up with what we saw with the larger fish that we were getting in the beach seines. Um, Buffalo Reef has the second highest catch per unit effort of any of these sites, with Traverse Point, which is that point that separates Little Traverse Bay and Grand Traverse Bay, having by far the highest CPUE. Now, I say CPUE, we know that this is not a quantitative indicator of catch per unit effort because we have no idea what the field of light is for, how far out we're attracting larvae in. So it's really difficult to say that that's a true measure of CPUE. So what we're calling CPUE is the average number of fish that we caught per hour at each of these stations. So this information is also uh, pretty interesting because this station here, which we're calling Burr, actually is on the black sand beach there at Buffalo Reef. And between 2008 and 2023, there's only been a handful of small age zero lake whitefish ever caught there. And by handful, I mean you could probably count them on one hand. So to get them in these kinds of numbers is incredibly interesting to us. This other site, which we're calling Gay, is also interesting because back in 1938, it was literally covered with 23 million metric tons of stamp sand. So in this aerial image from 1938, you can see the yellow box. That's kind of the general sampling location where we were when we were doing the light trapping. And you can see that gray swath of stamp sand extending about a mile and a half out into Lake Superior. If you look over at the image on the left, this is recent LIDAR survey data work done by Colin Brooks at Michigan Tech University, using drones and LIDAR data to track the movement of the stamp sand shoreline. As these sands are being transported downstream, this area is actually cleaned up, and all the stamp sand has been pretty much removed. So that green line that you see on the right, that's where the shoreline was in 2019. And then the yellow line that you see kind of in the middle of the figure, that's where the shoreline, the stamp sand shoreline currently stands in 2023. So it's moved about 150 meters in that 14 years. So, like I said, we're doing a lot of otolith-based work for these projects. So this is an image of one of Katie's tiled mosaics showing a larval lake whitefish otolith and then the different check marks. So this dark band here, that's going to be your exogenous feeding check mark. And then after that, you can see that there's these pretty equally spaced increments formed. So using that same kind of methodology that I explained earlier, we've been able to go through and back calculate their relative hatch dates at each one of these sites. And for the most part, they line up really well. You know, there's not much difference between Buffalo Reef and Grand Traverse Bay and at least four of the other stations in Keweenaw Bay. The one that does stand out is down at South Entry, which is a nice white sandy beach that's just off of the canal entry there on the Keweenaw Peninsula. We're not quite sure what's going on there, but we're going to certainly be sending in those individuals just to make sure that they're whitefish. And then we have some growth information that comes from this otolith work. I'm not going to focus on the age length relationships down on the bottom left, but just look at the growth rates on the top right. So again, it's kind of ordered the same way from left to right. You're going from the tip of the Keweenaw Peninsula down to South Entry on the right. Buffalo Reef and Grand Traverse Bay are right in the middle. And yeah, doesn't appear to be much difference in terms of growth rates during this early life stage between Buffalo Reef and Grand Traverse Bay, and at least Bay Degree, Little Traverse Bay, and Traverse Point. Um, but the fish that we collected at Gay have significantly lower growth rates, which you might expect. We know nothing about that habitat as the stamp sand is cleared away. We don't know if there's food. We don't know how much stamp sand remains there in the substrates. It's something that's just recently come on our radar and we want to look at. And then the growth rates were also pretty low down at South Entry. So again, we're concerned that maybe these weren't lake whitefish, but we won't know for sure until we get some genetic analysis on them. And then lastly, the last part of this, we use otolith microchemistry. We, as I said earlier, we were interested to see if we could discriminate fish produced at Buffalo Reef from fish produced at other reefs in the Keweenaw Peninsula. Um, And our primary hypothesis was that because copper is so high in the substrates and sediments at Buffalo Reef, that we should see a very unique copper marker in their otoliths, in the otolith cores in particular. Um, the problem with that is that there's a lot of copper in the substrates around Keweenaw Peninsula. If you look at Charlie Kerfoot's work from Michigan Tech, he has what he calls the copper halo, which is a halo of super high naturally occurring copper concentrations in the benthic sediments around the Keweenaw Peninsula. But what he also points out is that it's about 5 times higher at Buffalo Reef than it is in the natural background levels. So we were pretty hopeful that just based on copper alone, that we'd be able to use laser ablation, ICP-MS on the otolith core to answer this question. And so what I'm showing here on the bottom right is one of our larval fish otoliths where Katie took it down to the University of Windsor and used the laser to ablate just the core signal. And then we identified the actual core material by looking for the spike in manganese, which is known to occur in the vateritic core of Lake Whitefish. So this is kind of what the chemistry results look like. Kind of zip through this. So this is the manganese showing that there's no difference in the manganese across the 7 sites. These other elements, magnesium, zinc, strontium, and barium, when they're standardized to calcium, we don't really see any interesting differences in terms of their concentrations in whitefish otolith cores from the Keweenaw Peninsula. And again, the x-axis on this is laid out like it is in the others. Bay degree on the left is the northern part of Keweenaw Peninsula, and then South Entry on the right is the very southern extent of the peninsula in Keweenaw Bay. Then we have some elements that show some pretty interesting spatial variation. So we've got elements like chromium and nickel that seem to increase in the otolith as you move south along the Keweenaw Peninsula. And then iron and cobalt tend to decrease as you move south along the Keweenaw Peninsula. And then lastly, copper. There is a clear and distinct copper signal that relates to fish that we collected at Grand Traverse Bay and at Buffalo Reef, which is what we expected. So this is just kind of a quick and dirty non-metric multidimensional scaling plot showing the separation of these 7 collection sites. The first thing that you could see is that the Buffalo Reef and the Grand Traverse Bay, the 2 blue sites, kind of grouped together. And then there's a strong north-south gradient in terms of these sites along the second axis, with really the only big difference being the fish collected at Gay for some reason are lining up more as the southern population or the southern sites. When we look at our discriminant function analysis reclassification, so this is just showing the percent correct classification. I won't get into the details. The take home here is that we had pretty decent classification based on the 7 collection sites, with Gay being the one site that was really difficult to discriminate. But the interesting thing is that with the acceptance of one fish, the Grand Traverse Bay and the Burr sites, these 2 blue lines, they were always classified as one or the other and had a pretty high correct classification rate to begin with. So we decided, well, let's just compare stamp sand impacted versus non-stamp sand impacted. And when we did that, we get near perfect discrimination. So here the blue points are the stamp sand impacted. So that's Buffalo Reef and Grand Traverse Bay. And then the orange sites are the non-stamp sand impacted sites. So that's all the other sites. And when you look at our overall classification rate, we're at about 93%, with actually the stamp sand sites having the lower of the 2. So we can actually discriminate both the non-stamp sand sites pretty well and the stamp sand sites. So this is all pretty interesting. We were hoping we'd be able to do this, but to be able to discriminate fish at such a small spatial scale, I mean, the distance between Grand Traverse Bay and Buffalo Reef and say Traverse Point is less than 3 kilometers. So to be able to discriminate at that spatial scale is pretty interesting. So wrapping things up, some lake whitefish do appear to still spawn at Buffalo Reef, but it's in pretty low numbers based on our acoustic telemetry. This conflicts with what we see in November. Glyphwix gillnet surveys. We've been out there every year with them since 2019, and we catch a ton of gravid, ripe running female lake whitefish on the reef in November. So we're not sure what that means. There are very low numbers of age zero whitefish recruiting to the nursery habitat south of the breakwall. And even those that are recruiting there are probably facing kind of a gauntlet of potential stamp sand impacts because there's going to be low prey abundance and it appears to impair their growth rates. Larval hatch dates and growth rates are similar across the Keweenaw Peninsula, and they do compare favorably with other studies. And I'm speaking specifically to the work done by Aaron Dunlop recently. Um, and it's also pretty informative that our catch per unit effort and abundance is similar across those sites. Um, and I say similar because again, it's not really a true quantitative metric. And then lastly, stamp sand has a distinct chemical signal that we can use to discriminate among these potential reefs even at a small spatial scale. So what are the next steps? Well, if not here, then where and how many? So are adults spawning at an alternative site nearby, and that's what's producing the larvae that we're seeing there? Or are there just more fish spawning on the reef in that hotspot than we know? We don't have a way to tease that apart with the acoustic telemetry. So we need to develop other methods to complement the telemetry work. And then lastly, do larvae originate from another reef, another nearby reef? Doesn't seem like it. We've talked with Mark Rowe at NOAA about doing some flow modeling to see where these fish could have originated based on how old they are when we're collecting them. That may move forward in the future. It's hard to tell. And then lastly, can we use these archived otolith inventories that the tribes in the state have to retrospectively determine changes to the contribution of Buffalo Reef to past lake white fisheries in Keweenaw Bay? You know, specifically, can we identify fish that have this high copper concentration uh, from these archive otolith inventories, and then assign that to a Buffalo Reef fish versus a non-Buffalo Reef fish? Um, and then one of the questions that comes to mind for me is the copper that we're seeing in the otolith, does that reflect the environmental availability, which the literature says it does, or does it reflect maternal contribution? So were the females feeding in these copper contaminated habitats for some time, and then we're seeing that transmitted into the otolith through the yolk formation? And then, as you can imagine, there's a lot of people to thank on this, our partners, the funding sources. I could be here for the rest of the day trying to capture all of that. So I'll spare you that and just open it up to questions.
All right, Mike, that's a really impressive amount of work. I mean, you talked to us about telemetry and larval growth rates and zooplankton prey, and then otolith microchemistry, and going back to your title, sort of paradoxical. Yeah, there are some sort of paradoxical findings that you laid out. So as we open it up for questions, remember you can raise your hand via the participant list or pop your camera on, ideally. You can also write a message in the chat that we could pass along to Mike. So maybe I'll just open it up, Mike. So it's just so I didn't misunderstand anything. It's clear from the harvest that catches are down in that region. And the culprit was believed to be something early life history associated with the spawning reef.
Mhm.
You weren't really seeing anything with respect to larval densities with the light traps. Maybe you did see some, or the growth rates for the larvae that were sort of surprisingly low. But when you sort of looked at the beach staining for some of the early juveniles, you did see lower catch rates
Yeah.
nearby Buffalo Reef, and they were growing more slowly, potentially associated with unexpectedly low densities of invertebrates maybe they were feeding on. Is that sort of if there's a bottleneck, it may be more sort of between the larval and juvenile stage? Is that kind of the one of the hypotheses, or is it just too soon to kind of know what the impact might be and what the mechanism is?
I do think that that's one of the bottlenecks. Um, this abundance of larvae that we're seeing on the black sand beach, there's almost no zooplankton for them to feed on once that yolk sac is gone, right? And they're most likely being entrained in that area because the flow dynamics with that jetty wall there. So they probably recruit there and then they don't find anything to eat and they're just perishing, right? The other bottleneck, and I didn't talk about it today, is and it's stuff that I've presented elsewhere, is that we've been doing experimental exposures of developing lake whitefish embryos. That's where our laboratory reared fish are going, or that's our source for that. And basically, what we see is that when you expose them to increasing levels of stamp sand during that critical development period, you see pretty high mortality rates once you get above 30-40% stamp sand. So your odds of an embryo successfully hatching are near zero when concentrations are greater than 50% stamp sand.
Yeah. So, another thought is, I mean, in Lake Ontario, some work by Brian Weidel and Alex Gatch and those are looking sort of at the interstitial spaces.
Mhm.
And so you're looking at sort of the toxicity of those interstitial spaces and to what extent is there interstitial spaces filled up by stamp sands versus sort of normal sands? And is that just a natural process where, I mean, maybe the stamp sands are lighter and more prone to being sort of swept into the reefs, but it didn't look like it was 100% stamp sands pretty much anywhere. So tell us about sort of that infilling of sands, some of them stamp sands, into the Buffalo Reef and sort of that effect of reduced interstitial spaces that could also be beyond the sort of the toxicity that you just mentioned.
Sure. So we are quantifying the amount of sand in these interstitial spaces to pair with those water samples that I talked about. So we have estimates of percent stamp sand. We're using a suction sampler. If divers go down and they basically move the rocks out of the area where we took the water sample, and we use a suction sampler to lift all of that sediment up to the boat, and then we send it off to USGS Circ Lab in Colombia to get a full analysis of the heavy metals in those sediments. And then we do some stuff in the lab to estimate, grain size, the amount of stamp sand that we're collecting per gallon of flow. Um, so we do have some indication, and so far what we've found is that there's stamp sand in just about every single interstitial space that we've sampled. Some are much worse. The ones that are near shore, as you would expect, are much, much worse in terms of infilling of those interstitial spaces. Part of the problem is that stamp sand does not, as you kind of alluded, is stamp sand does not behave like a natural sand grain. It's actually more dense. So once it sinks into the interstitial space, I think it's probably just hard for natural processes to move it out. So we suspect that our stamp sand estimates from the suction sampling, we suspect that they're probably on the low end for the estimates because of the way that the stamp sand sinks down into the interstitial space. One thing we don't know is how much interstitial space is there and how much is free. Um, We've recently found that there's a clay layer on some parts of the reef, and we don't know if this is a natural clay layer or if this is at one point when they were processing these stamp sands, they were producing what was known as a slime. So it's the really fine particulates that they were basically combining with water and washing out into the bay. Um, and that's sitting underneath the interstitial space between the rocks and the limestone. And it is 10 times higher in copper concentration than the actual sand grains themselves.
Like, hopefully people will add something here. But I mean, so what about Gay? You talked about Gay. You had an image there from the 1930s of inundation of stamp sands there, and now that's one of your sites. And you didn't really talk about it being stamp sand impacted now. How does the Gay site and the historical stamp sand sort of poisoning, if you will, sort of, how do you relate that to what we're seeing today at Gay? Has it sort of,
Yeah, we don't know. That's part of the problem. We only recently it, the site at Gay was only recently brought to our attention last summer. So we
hmm,
that's when we started sampling there just to start developing some sort of baseline for that. I
hmm.
think the interesting part is that it gives us a kind of a recovery site to monitor because it's been cleared of stamp sands by natural processes. Do we start seeing benthic invertebrates come in there? Do we see fish actively feeding and using those habitats? With the larval fish, it's hard to say that they're actively settling in there. They're just, they're being transported from someplace upstream, right? Um, but we don't know anything about it. This was the first time someone's taken a biological sample there that I'm aware of.
So some of those sands, you said it's been sort of recovered. Some of those stamp sands have moved down into Buffalo Reef. Is that the sort of hypothesis? They've been sort of, yeah, okay.
Yeah, it's been exacerbated by efforts to kind of maintain the beachfront. So as they're mechanically pulling these things back and dredging out the trough, it creates space for the stamp sand to go. So we think that that's kind of led to all of that eroding faster and moving downstream.
Okay, cool. Hey, Scott Niels, let's kick us off with somebody other than Bo asking questions. Thanks.
Thanks. Yeah, great presentation, Mike. I was thinking you mentioned about entrainment a little bit ago with the jetty, and I was just curious if anybody has done any modeling work or any work with the currents and modeling the larval drift in that area. And specifically, I was just thinking about whether or not you might see source sink dynamics with all the other potential sources of production that are right around that area.
Yeah. No, I kind of alluded to it. Andrew Muir and I kind of started conversations with Mark Rowe at NOAA about doing some of this. Um, because we're also interested how that dynamic affects the movement of stamp sand onto the reef. Um, so it's kind of a double whammy in terms of the flow dynamics there in the nearshore area. What I can say is that the state and KBIC are putting forth effort every year to remove sands from that exact same area because every year a major storm comes along and that's where it all ends up, at least the stuff that can be moved. It all ends up right there in that spot. So they go in and they mechanically pull it back from the beach and create another hole, right? And then the next storm comes in and does the exact same thing.
Thanks, Scott. Brian Weidel.
Hey, Mike, great talk. Thank you for sharing.
Thanks,
Like
Brian.
Bo said, impressive amount of work. The you mentioned, I think I heard this right. The Buffalo Reef area, you are estimating it at like 33 hectares. Is that right?
Is that 2,200 acres?
Pretty close.
Can't do that math.
Yeah, two and
It's
a half.
close.
Yeah,
I think so.
Yeah.
but like what I was more interested in sort of the heat map from the telemetry results in those 2 points. Just ballpark, how big are those?
The critical core area, so we estimated this for the Army Corps. The critical core area is about 500 acres.
Okay. All right.
Yep.
Still pretty big.
It's still pretty big, and then a lot of that I think has to do with, it's so few fish, right? If we had 30 or 40 fish going in there and spawning, we might be able to narrow that search area down a bit more.
Right. Okay. Yeah, yeah, yeah. I think I misheard maybe 33 square kilometers. That makes more sense. But okay, it's still pretty big up on the tops of those points. Just trying to Superior can sometimes give you some difficulty with scale, as I'm
Yes.
sure you handle all the time. So neat stuff. Thank you.
Thanks, Brian.
The other thing about the telemetry data, Mike, I thought was interesting and thought might show up later in some of the larval work or the beach staining was the higher concentrations. I think you showed up at the north, like beta Greece, and yet you didn't seem to show any higher, larval or sort of juvenile catches there. Is that true?
That is true.
Okay.
And then I think it should be noted that the beta degree is one of the most consistent producers, whether we're
Hmm.
talking about at the larval stage or the age zeros. Um, so you're right. It will be interesting to see if we continue this kind of long term with the larval monitoring, and we can come up with a better estimate for, a quantitative estimate of larval density or catch per unit effort. Just how important beta degree is. There is a small stamp sand pile up there, but it's inshore in the on the inland water body, so it's not really a concern.
Yeah. Brian, another question?
I might have missed it, but the as you're tracking these fish, year to year variations of the individual, are they
Mhm.
going back to the same places?
Yes. So in all three of those heat maps that I put up, there are two fish that we tagged in 2019 that have returned every year. And then as we introduce the new cohort of tagged fish, we get some individual variability there.
Any any male to female pattern in that?
They've all been male. Well, those are male. So the fish that we tagged in 2019 or in 2022 and 2023, we tagged them in the spring, so we couldn't
Uh
reliably sex them, right?
yeah. Huh. Damn. Interesting.
So, Mike, what about you mentioned lake trout? Of course, I know you've been doing some other studies, and then you mentioned sort of the can't rule out the possibility of Cisco or a later cohort of hatching, or maybe some smaller larvae than expected. Do you have a sense of like more the community use of, say, Buffalo Reef or other areas, and sort of I don't know, maybe tell us a little bit about the lake trout part of the story, and also from local sort of tribal knowledge or commercial fisher knowledge, sort of the use of Cisco in this area too, and how Cisco might be impacted or not?
Sure. So in terms of lake trout, they are they continue to actively spawn right on the tops of those two humps in the middle of the reef. So there seems to be spawning activity on the top of each reef. It's fairly consistent, but it's limited to the fish that we tagged when they came onto the reef to spawn in 2019, because when we were tagging the whitefish in 2022 and 2023, we tagged about 100 lake trout as well. And those fish appeared to be avoiding Buffalo Reef. They come out into kind of a hot spot on the southern part of the reef. We don't know if they're spawning there or not. We recently expanded the VPS array last year to kind of get at that, and we're still trying to dig into that data. Um, in terms of Cisco, we have seen in our at least in our seine nets, especially that 2022 year class,
Oh
2021
yeah.
years. Yeah, we saw a lot of much smaller coregonid larvae. We'd catch them sometimes, but for the most part, we could see them getting out of the net as we came in to the beach. So we do have those samples, and we've considered, trying to get the genetics analyzed on those as well. And I do know that historically that there were Cisco catches at Buffalo Reef. And I've seen some images from a USGS report in the 50s showing a big haul of Cisco on one of the Peterson's fishing boats up there. But I don't know. I don't think there's been much there recently. I don't recall seeing many Cisco on the reef in any of our gillnet samples.
Okay. Thanks.
Yeah, I could probably add to that. There's not a big Cisco fishery in that area. There has been some Cisco fishing down more south in the bay, but not up close to the reef there. Yeah.
And that's like long-term, Bill, or that's just more recent, or
No, in the
say?
past, it's more been in the past. There hasn't been a lot of targeted Cisco fishing in that area since
Mhm.
the 2000s. Yeah.
Yeah. Okay. Yeah. Thanks, Bill.
And I will say that I've been working with folks at Michigan Tech, one of whom Travis White happens to be a fishing guide up there. And this year lake trout have been their stomachs off of Buffalo Reef have just been packed with what look to be Cisco's or Kai in that 4 to 5 inch length.
Okay. Eric Olson.
Hey, very good presentation, Mike. Thank you. Interesting.
Thanks.
Just out of curiosity, in your telemetry work, how much overlap, if any, did you see between whitefish and lake trout on the spawning grounds?
So based on
Timing-wise specifically.
what's that?
Timing-wise specifically.
Yeah. So based on the two studies, based on the VPS study, we don't see a whole lot of overlap on Buffalo Reef. There might be a few stragglers of lake whitefish that come on early and a few lake trout that hang around a bit longer. But there's just not a lot of overlap there. If you look at the larger array and the individual reefs, when we compare that, we see that there are lake trout and lake whitefish present on each one of those reefs, between the 1st of October to the 1st of December. We don't know spatially how much they overlap, but they're probably within a kilometer of the receiver. So it's hard to say that there's much interaction at that point, but in the VPS array, they don't seem to interact much.
That's what I was hoping to hear. Thanks.
Mhm.
All right. Thanks for that question, Eric. Any more questions for Mike?
I'll ask a question.
Yeah, right.
Hi, Mike.
Hey.
Great talk. Really interesting stuff as usual from you. It's pretty cool. I'm just wondering if you've looked at or have started to think about maybe some other differences among the habitats, whether they're interacting with the stamp sand itself, or maybe indirectly influencing the fish. So, for instance, like if the stamp sand alters the phytoplankton, or as that could be reducing or stunting the food web. And yeah, if you just thought about that at all, and if maybe those are some other things that could be going on.
Yeah. There were a couple proposals out of Michigan Tech a few years ago to look at like the periphyton that accumulates on the reef in the summers. And we have collected some of that and sent it off to Daniel Cleveland at USGS CERC for the analysis. We just haven't seen it back, seen the data from that just yet. But we do suspect that base of the food web is probably contaminated based on how much stamp sand is in those environments where we see it growing.
Yeah, could imagine that's probably true. I also had just a follow-up question too, or a comment, I guess, about light traps, because obviously no shocker, I'm thinking about the water transparency. But do you have any information or relative idea of are there big differences in the transparency between those sites within just kind of short range there? Or are they all kind of relatively the same? Because I kind of would have thought, I guess I wouldn't have even thought of thinking about that with a light trap.
Yeah.
It makes sense. I don't know why I wouldn't have thought of it, but
They're relatively the same, mostly because we try to go out in nice calm conditions. So and we need clear water so that we can actually see the larvae, right? If we're doing the active light trapping. I will say that the active light trapping, the lights we're powering those with a couple of Jackery units. So they're super bright. So they have a nice big bright field to bring in fish from fairly far away. When you compare that with the light traps that we made, that light just doesn't penetrate very far out. They're low level LED lights that are stacked inside, and we did have some success. We did get larvae, but for the most part, what we'd notice when we were out there at night is we'd do kind of a slow drive by on those, and we'd see the fish swimming around and think, all right, well, when we pick these up, we're going to have a lot of larvae in there, and then we'd pick them up and the traps would be empty. So we don't know if they're getting into the traps and they're able to get out, or if they're just not entering at all. But what we do know is that sticklebacks enter the traps somehow. We don't know how because the gap's only about a millimeter and a half wide for them to enter, and we had tons of sticklebacks in those traps. I think that they just dart in and wedge themselves in.
interesting.
Huh. Thanks, Nikki. All right. Well, if you're still on, as like almost 50 of you are, you can give sort of a virtual clap or use one of your emotions through Zoom to let Mike know you appreciate that great presentation he gave. And we look forward to seeing many of you back for another Whitefish Talk, talking about the genetics by Jared Humala in about a month. Thanks again, and have a great weekend, everybody.